Okay, welcome back, everyone. So I hope you had a chance to get a little break here. So we're going to continue maybe about 30 minutes of conversation from chapter 2 to really kind of wrap things up. So we're in a good starting place for chapter 3 next week when we're physically back on campus together. And so just to let you know, I am recording this portion of our lab time because I'm using it to explain content from the textbook. Once we get past that, I'll stop recording. We'll just do our normal lab stuff, okay? Do you have any questions before we continue? Okay, all right. Well, let's continue then. I'm going to share my screen with you. And, from 35, 40 minutes ago, we just started talking about meiosis. But I'm about to give you some descriptive text of meiosis 1 and meiosis 2. Before I do that, I think it probably would be good for us to see a cartoon on this, just so you have something in your mind to think about while we're talking. And by the way, the first video I shared with you on mitosis was unfortunately a little blurry, and I apologize for that. Wanted you to know that I found the same video on YouTube from a different provider, and I've substituted that updated link in your notes, so the next time you go see it when you're studying, you should get a nice crystal clear video, okay? All right, so let's get started with the meiosis video, okay? So the meiosis video, I think, is going to do a little compare and contrast with mitosis, and that can be useful when we're talking about these topics. So let me get started here. Okay, this will be about 6 minutes, so relax, enjoy it, because you can watch it again during your study time. And here we go.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Okay, folks. So hopefully that puts us in a good shape to talk about this and give you a skeletal outline of these things. Obviously, your textbook is your reference. That's where you really want to dig into things. But we need to be sure that we're talking about these things as well, highlighting features that might be, that perhaps you may miss when you read the book. Do you have any questions about the video? Okay, well, again, the video is linked here for you, so let's get started. We're going to talk about meiosis 1 and meiosis 2. And so meiosis 1 and meiosis 2 are graphically depicted for you in figure 211 of your digital textbook. The textbook does certainly a better job than I could draw for you, so I'm just going to let you look at that while we're having our discussion here. But let's talk about this. Okay, so let's talk about meiosis 1. Okay, meiosis 1 is going to be made of, prophase 1, metaphase 1, anaphase 1, telophase 1, followed by its own round of cytokinesis. This I don't think is a surprise to you. I mean, you've had this in biology 101 or 102. However, it might be a little bit of a surprise to you to know, and this is something that your textbook is not going to tell you, just like science has advanced to the point that we now in mitosis, we don't just talk about prometaphase and metaphase, excuse me, we don't talk about prophase and metaphase, we have prometaphase, we kind of subdivide even the sections more narrowly. Well, in meiosis, geneticists are realizing that there are defined substages within prophase 1, okay? So I'm not going to give you a lot on this, but I do want you to be aware of it, that we're advancing our knowledge. We're starting to understand things more deeply than ever before. Okay, so even the things that I'm about to mention to you are not formally mentioned in your textbook. We'll keep this nice and simple, but I do want to make you aware of it. So we have prophase 1, which itself is made of 5 substages that geneticists are looking at. The 5 substages of prophase 1 are leptonema, zygonema, pachynemia, diplonema, and the 5th and final substage of prophase 1 is called diakinesis. So I get it, if you're not going to be a scientist, if you're not going to be a researcher, you're kind of like, well, you know what? I was quite okay with not knowing those words, right? Because they're big and obnoxious, aren't they? So don't worry, this will be a very gentle introduction to what I'm talking about here. Your textbook does not give you these names anywhere in chapter 2. In fact, I looked in the index and that they're not in the index of the entire textbook. But I do want to highlight something. And so what I'm going to try to do is bring a picture into your Mathematica document for you. Okay, so let's do that and see how that looks. Okay, there we go. Okay, so there you see leptotene, which is a different way of saying leptonema, zygotene, different way of saying zygonema, pachytene, pachynema, diplotene, diplonema, diakinesis. Okay, so let's talk a little bit about what's happening here in each of these substages of prophase 1. All right, so I'm going to continue here. All right, leptonema. Probably the most important thing you take away from leptonema is that during prophase 1, leptonema is a specific substage in which the chromatin condenses into chromosome. Okay. Now, when the chromatin condenses into the chromosomal state, you will be able to see bead-like structures that form along the chromosome. And those bead-like structures are called chromomeres. And you can see that in your textbook. Take a look at figure 2.10. Take a look at figure 2.10. Okay. When the chromatin condenses, when the chromatin condenses, bead-like structures form within the chromosome called chromomeres. Okay. And again, I reference figure 2.10 for you on that. So if you look very carefully at figure 2.10, the most leftward picture there is showing you the bead-like appearance of these chromomeres that are positioned along really condensed regions of the chromosomes. You know, truthfully, we don't know exactly what those things are doing, but we can see them in the microscope, right? We can see them in the electron microscope. Okay, so also during this period of time called leptonema, there's a molecular process that's taking place, and it's called homology search. Now, we won't attempt to explain the molecular details because honestly, science does not understand the molecular details yet. But during leptonema, this process called homology search is happening. And the purpose of this process called homology search is to help the cell locate the homologous chromosomes, right? Because one of the things that's very different between meiosis and mitosis is, in fact, the idea that the homologous chromosomes actually align next to each other in meiosis 1. That never happens in mitosis. At least if it did happen, it would be random. But in meiosis, it happens intentionally. Okay, so let me put a couple words in your notes, okay? During leptonema, a poorly understood molecular process called homology search begins, okay? The purpose of this homology search is to enable the cell to locate and orient and bring next to each other the homologous chromosomes. The purpose of homology search is to allow the cell to locate and properly position homologous chromosomes next to each other. Okay, so again, that homology search process is unique to meiosis. It does not take place in mitosis. Okay. All right. So that's the first stage, leptonema. Chromatin condenses into chromosome. You find these bead-like structures called chromomeres that show up in the electron microscope. We are aware that there's a process called homology search, which the cell is engaging in through machinery that we don't completely understand yet, that's doing something to target and identify homologous chromosomes and bring them into physical proximity next to each other. Okay, next. The next stage is zygotine or zygonema as I learned it. Okay, with zygonema, the chromosomes that exist, they are going to condense even further and become shorter. Okay, so the chromosomes are going to shorten in length. Okay. And during zygonema, you're going to find that the homologous chromosomes, they start to get into position so that they can come together in synapse. Now, the synaptic process of bringing them together does not complete in zygonema. It gets started, but it doesn't finish till the next substage, okay? So during zygonema, you're going to have a multi-protein machine, if you will, called the synaptonemal complex that's going to kind of position itself, it's going to nudge itself in between the homologous chromosomes and start moving them around and fine-tuning their position. Okay, so a multi-protein complex called the synaptonemal complex forms. Okay, it literally forms. What it means, the synaptonemal complex is made of many proteins that layer on top of each other like a giant three-dimensional jigsaw puzzle. So this protein complex that we're calling the synaptonemal complex, it forms because these proteins start laying on top of each other, and it forms in between the homologous chromosomes so that it can help facilitate the final stage of synapsis. So it forms in between the homologous chromosome pairs. Okay. And so it's important to geneticists. I don't know that it's important to most undergraduates, but if you really want to understand this process, right, you want to know as much as you can about it. Researchers need to understand as much if we're going to figure out aberrations in the process like cancer. It turns out that when the synaptonemal complex is doing its thing, typically most textbooks, you'll see this. You'll see where the homologous pairs are kind of drawn next to each other as though they align that way. It turns out they don't really align next to each other. They kind of more align on top of each other. Okay, so it's hard to draw that in a textbook because if you show two things overlap, students might miss the fact that there's really two things there, so they separate them to show them this way. And I would draw it this way too because I'm not much of an artist. But the reality of it is, in the living cell, the homologous chromosomes aren't so much next to each other as they are on top of each other. Okay, so let me take note of this. Homologous chromosomes are positioned in a way that they are more on top of each other than side by side. Okay. So at this point, it would be difficult to interpret this from a traditional textbook picture or textbook drawing. Okay. Let me say this to provide some context to you. Okay. During the zygonema stage, the synaptonemal complex interacts with the homologous chromosomes and positions them roughly on top of each other, and the distance in between them, the distance separating the homologous chromosomes, is on the order of about 300 nanometers. 300 billionths of a meter is all that's separating those two structures. Okay. All right, so the distance is roughly 300 nanometers. Okay, so that's what happens in zygonema. Zygonema, the DNA, the chromosomes shorten in length, the synaptonemal complex forms, and it starts tweaking the position of the homologous chromosomes that they're roughly laying on top of each other, separated by a distance of around 300 nanometers. Okay. Next. Next, we have pachynemia. That's quite a word, isn't it? Pachynemia. During pachynemia, the homologous chromosomes complete the synaptic process. So the chromosomes actually synapse, which means they join. Okay. So homologous chromosomes formally synapse. And what that means from a practical perspective is this, that at zygonema, the distance between those homologous chromosomes is about 300 nanometers. When the chromosomes synapse, the distance between those chromosomes becomes 100 nanometers on average. So we're really compacting these structures and getting them very close to each other physically. Okay, so the distance between homologous chromosomes shortens to about 100 nanometers. Okay, which is not a lot of space, folks, right? And so it is in the pachynemia stage that crossing over is going to take place, because synapse has occurred. The strands of non-sister chromatids are close enough together. They're lying on top of each other. They're only separated by 100 nanometers. They're close enough together that the enzymes that will encircle those non-sister chromatids can go in, snip segments of DNA, swap them back into position, glue them back down, and now we have homologous chromosomes that have different non-genetically identical chromatids. Okay, so it's going to be during pachynemia that crossing over takes place. But, right, there's always a giant but. Pachynemia is the stage that crossing over takes place, but because of how close the chromosomes are lying on top of each other, you would not be able to notice that crossing up. You would not be able to visibly see crossing over happening in the microscope until you get to the next stage, which is diplonemia. So pachynemia, crossing over takes place, but you can't see it until you get to the next stage of prophase 1, which is diplonemia. But crossing over is not visible in the microscope at this stage. Okay, so and please understand, we're talking about structures that are way too small to be seen with a light microscope. The only way you have a chance of looking at this stuff is with an electron microscope. And even with the standard electron microscope, you can't make these things out. Okay, it's super mega small. So, okay, so pachynemia, the homologous chromosomes synapse, which means they get very close together, 100 nanometers distance. Crossing over happens, but you would never see that under the microscope. The realization that crossing over happened in pachynemia had to be figured out by other laboratory processes, not using microscopes, but by using radioactive tags to see where the radioactivity is moving and stuff like that. That's the only way we know this, okay? So let's continue. All right, diplonemia. Okay. So again, diplonemia is the next stage in which in the microscope you would think that crossing over took place, but really it took place during pachynemia. Okay, so during diplonemia, this is the first stage. This is the first substage of prophase 1, where you can visibly see the result of crossing over in the microscope. Okay, so we have had the exchange of genetic material at pachynemia, but we can't witness it in the microscope till diplonemia. And then finally, substage 5, diakinesis. During diakinesis, the nucleolus and the nuclear envelope break down, right? We said this during our discussion of mitosis, that the nuclear envelope has to break down so that the spindle fibers from the centrals can lock on to, the chromatids. Well, that's going to happen too. We're just giving it a different name. We're saying it's diakinesis in prophase 1 of meiosis 1, that the nucleus and the nucleolus inside the nucleus kind of just disintegrate away. Okay, they lose their structure. Okay, the nucleolus and nuclear envelope break down. Okay, we could also say it's at this stage that the spindle fibers attach to the chromosomes. And from that point, we're moving well into the start of metaphase, so that the chromosomes will be positioned along the metaphase 1 plate. Okay. All right. So folks, this is really, these prophase 1 substages, I don't want you to build a lot of anxiety in yourself about them, but this is information that is not in your textbook. So, there probably will be a question or two coming from it on a future exam, since I'm spending class time on it. Okay, so please be sure that you're at least thinking about this a little bit. Do you have any questions right now? Okay, well, hopefully you're not passed out because I can't see you. But let me just give a little bit of verbiage to some of the rest of the stages of figure 211 in your textbook, and combined with this information, the notes, the reading, the videos, doing some of your homework in mastering genetics, I think you'll be quite in good shape. Okay, so let's continue here. Okay, so we talked about prophase 1. Let's continue with metaphase 1. And that's going to be, excuse the scrolling back and forth, that's going to be letter B. Okay. So metaphase 1. Metaphase 1, the homologous chromosomes, which are now made of non-sister chromatids, they're positioned at the metaphase plate. Okay. So I have a question I'd like to ask you now before I kind of give you the answer by giving the notes down. So let me type this question in the chat window. Here we go. During meiosis, do cells become haploid after meiosis 1 or after meiosis 2? Okay. All right. Thanks for your feedback, everybody. I appreciate that. And so here's what I'll say. Reevaluate your answer. Reevaluate your answer as we go through this process, okay? All right. So it's actually at the end of meiosis 1 that cells become haploid, okay? Because remember, haploid means you have one copy of every chromosome, okay? All right. So let's continue. Letter C, anaphase 1. Don't worry, folks. Well, maybe you should worry, but don't worry. We're going to do some activities. We're going to at least start some activities today in lab that are going to be a lot of work. I'll be honest with you. It's going to be a lot of work, but it should enhance your understanding of these processes. anaphase 1, okay. So when you look at a typical drawing in figure 211 and you look at metaphase 1, I guess I should draw a little bit. So let me ask you this, folks. Let me ask you this. I'm going to ask you a couple questions in the chat window. How many chromatids does a monad have? Okay, I'm not getting an answer, so maybe this is something we need to talk about. Okay. Yeah, it has one. That's right. A monad is one chromatid. Okay. And just so you know these things, a dyad equals 2 chromatids. Okay. A dyad equals 2 chromatids. So folks, take a look at, let's see. Let me bring something up on Google here and Google Images and see if that'll work. All right, let's take a look at this. Okay. Come on. Not what I want. Don't do that. Okay, let's try this. We probably should talk about this. And this all feeds into our theme today of cell cycle and meiosis. So let's take a quick break here on my notes from what I'm describing and talk about terminology. Ooh, nice and big. How about that? Okay, good. All right, now. All right, so you appreciate, some terminology here. And let's see, let's see if I can draw on this thing. Okay, so you appreciate that this right here, these two pink things is one dyad. Okay, these two blue things here is one dyad. So when you have both of these things together, here's a pair, one pair of dyads, one pair. A pair is 2, right? So one group of 2, one pair of dyads, which is the same thing as a tetrad. Same thing as a tetrad. Okay, question in the chat window. Here we go. How many chromatids does one tetrad have? Okay, good, folks. Good, it's got 4, right? So you're definitely going to want to be familiar with the phrases monad, dyad, tetrad, because your old genetics teacher is going to use them, okay? So this one pair of dyads, which is a tetrad, you can see in this drawing is one pair of homologous chromosomes. Don't know why my computer insists on making square letters, but hopefully you can interpret that easy enough, okay? All right, so do you have any questions about the terms monads, dyads, or tetrads? Okay, all right, good. So hopefully that clarifies a little bit some of this language that gets thrown around. And so let's continue here. Talking about, see, we were at metaphase 1. Let's go to anaphase 1. Okay. And I'm sorry for going back and forth here. Anaphase 1 is going to be letter C. So with anaphase 1, you know, what the heck do we observe? Okay, during anaphase 1, the two bivalents. Okay, so that's another word, isn't it? Okay, I need to add that to your list here. Hold on, folks. It's just that there are terms which are synonymous, but if you don't know that, it might throw you off. Okay. So let's do this. Okay. Okay. One dyad one dyad is the same thing as one bivalent. Oh, that's horrible, the way this thing draws. Okay. I really don't draw like a kindergartner, folks, but this is how it's going here. Okay, so let me ask you this. How many chromatids does a bivalent have? Okay, good. It has 2. A bivalent is the same thing as a dyad. A bivalent is the same thing as a dyad. Each term refers to 2 chromatids. Okay. When you have 2 groups of dyads or 2 bivalents, you're going to have 4 chromatids, which is the same thing as a tetrad. A tetrad is the same thing as 1 pair of homologous chromosomes aligned next to each other. And so you need to appreciate this language if you're going to understand reading your textbook or reading your instructor's notes, okay? All right, during anaphase, the 2 bivalents, right? So here's a red bivalent and a blue bivalent. During anaphase 1, the 2 bivalents of each tetrad start to get pulled to opposite poles, right? The 2 bivalents, or you might say dyads, of each tetrad are pulled to opposite poles. Okay. And so let's put this as a question in your notes here. When the 2 bivalents separate, what is the fancy term for that separation? What do we call that separation? It's called, what's the fancy term for separation of the bivalents? It would be the same term when we're separating individual chromatids during anaphase of mitosis. It's going to be the same term. Okay, let me push you forward. It's called disjunction. It's called disjunction. Disjunction means they separate. Disjunction means they separate. So you may have heard in other courses that sometimes genetic diseases and unusual things happen when a cell experiences non-disjunction, right? Trisomy 21, Down syndrome. When the bivalents do not correctly separate, that's non-disjunction. And it can cause a cell to have more than the normal copies of the chromosomes, such as Down syndrome children experience. Okay. Let's continue here. I don't want to get too much longer, but I do want to finish this. So telophase 1. Okay. Telophase 1. This is the end of meiosis 1. To be completely honest with you, telophase 1 does not happen in every type of cell on the planet that's been observed. Some cells perform telophase 1, other cells don't perform telophase 1. So I'll put it this way: doesn't occur in all species of cells. Okay. Those that do perform telophase 1, when telophase 1 occurs, typically you're going to temporarily reform the nuclear membrane. Okay. So I know I'm going kind of quick here, folks, but really I do want to get to the lab content, but I also want to give you the notes. So let's finish this out, and we'll pause for questions and things. All right, so that's telophase 1. That finishes meiosis 1. So at the end of meiosis 1, we have introduced the genetic variation through crossover and also the random alignment of the homologous chromosomes on top of each other at the metaphase plate. So the mechanisms of genetic variation have now already taken place. They've already happened in meiosis 1. So as my earlier question alluded to, in terms of do the cells become haploid at the end of meiosis 1 or meiosis 2? Well, they do become haploid at the end of meiosis 1. It's just that now each of the chromosomes has twice the DNA because it was copied during the S phase, which means those haploid cells at the end of meiosis 1 have to go through another round of cytoplasmic division, so I can create 4 haploid cells that have the normal DNA content of 3.1 billion nucleotide pairs of DNA. Okay, so let's continue here into meiosis 2. All right, so I just want to try to keep my notes a little bit organized for you. Okay, so step 6 here. Meiosis 2 in some cells is preceded by a short interphase period where no additional DNA is copied. Okay, no additional DNA would be copied if there's a short interphase, because that would really throw things out of whack. Okay, so let's continue. We have prophase 2. And again, you can be looking at figure 211 in order to understand that. So at this point, cells are haploid. Okay, at this point, cells are haploid. The chromosomes exist as dyads. Okay. And the chromosomes that I'm talking about, the chromatids that make up those chromosomes, are not genetically identical because of the crossing over that happened earlier in meiosis 1, specifically pachynemia of prophase 1. Okay. You know, it's moments like that you start thinking, why the hell did I take this course? Okay. But, you know, somebody has to know this stuff, don't they? So, okay. That's prophase 2. Metaphase 2. Metaphase 2, the dyad chromosomes, the bivalents, not the tetrads, the bivalents, the dyad chromosomes line up at the metaphase plate. Okay. Third stage here, anaphase 2. Okay. The non-sister chromatids of each dyad separate. They get pulled towards the opposite poles of their respective cell. Okay. Telophase 2 will take place. During telophase 2, the nuclear envelope will reform. And of course, for the cells to actually physically separate, you know, telophase 2, whether we as human beings consider cytokinesis a part of telophase 2, or we think of it as its own thing outside telophase 2. Again, the experts disagree on how you organize this. Who honestly cares? You understand that for telophase 2, the envelope has to form, and then for the cells to separate, the cytoplasm has to separate. So that's going to happen in this period of time too. That has to happen, right? So cytokinesis occurs, division of the cytoplasm, and voila, we are now left with the 4 gamete cells that you see on figure 211. In my book, it's page 27. You see the 4 haploid cells, you know, which would be sperm or egg, okay? 4 haploid. When I say haploid, I'm talking about N equals 23. 4 haploid cells are produced. And of course, you know, how the sperm and the egg come together is somewhat random as well. So this generates tremendous genetic diversity in terms of, you know, the kinds of children and the characteristics and abilities and features and traits that they would possess. Okay, so that's a lot, folks, and I think I better stop because I'm probably causing you to get sleepy here. So any questions at this time? Okay. If you don't have questions, then we'll transition into our lab time together, okay? So I'm going to stop recording. At this moment, I'll stop recording, and you know, today's lecture recording and notes will be placed in Mastering Genetics. If not by the end of tonight, then tomorrow I'll get them there.